Very much, Dan, for the introduction, and welcome everybody to this webinar. And today we're going to hear some diverse approaches to ensuring that published outputs in academia are robust, reliable and transparent. And I'd like to thank Mark as well for putting this webinar together. It builds on a UKRN event that we had around about 2 years ago on the potential for total quality management approaches to improve the quality of academic outputs. And Mark contributed a presentation back in 2023 on a planned reproducibility initiative. So it's great to have him back today to hear some of his insights. In addition, we're also delighted to have Dr. Catherine Winchester, who will be sharing the approach which is embedded in CRUK Scotland. But first of all, we're going to hear from Dr. Nick Beazley-Long on his journey on getting to grips with FAIR principles and how these can enable reproducibility checks. Nick is a Knowledge Exchange Fellow here at the University of Bristol in the School of Health and Life Sciences. He's got a research background in the role of microvasculature in pain from a PhD that he did in physiology, pharmacology and neuroscience here at Bristol. He's also spent some time in commercial research environments. I think Boehringer Ingelheim is one of the companies he's been working at. And his passion is helping researchers to build transparency and reproducibility into their research processes to help combat low levels of reproducibility and trust in academic research outputs. So, Nick, over to you.
Okay. Can you all see my screen? Yep, thumbs up. Great. Okay, so thank you. Good afternoon, everyone. Thank you for joining this webinar, and thank you to Mark and the organisers for giving me the opportunity to introduce my FAIR journey to date. I'm Nick Beazley-Long, a Knowledge Exchange Fellow at the University of Bristol, and I work across the faculty, the new super faculty of Health and Life Sciences. So this talk is actually going to be more of a sort of chronological record, if you like, of my FAIR journey. When I started as a Knowledge Exchange Fellow a year and a half ago, I wasn't very FAIR savvy at all. So I've been learning along the way. And it all started with a manuscript statement and data availability check that we did for articles that came out of Life Sciences, the Faculty of Life Sciences here at Bristol in 2023. I'm going to introduce some of those results. This then led to a survey we ran in investigating barriers to data sharing across the faculty. I'm also going to introduce some of the FAIR checking tools that I've experienced. The Australian Data Research Commons FAIR self-assessment tool, and also an automated FAIR checker that's come out of the French Bioinformatic Institute. Then I was asked to do a 30 article FAIR check for the psychologist, and I will briefly introduce that. And then hopefully by the time we've got there, I will have convinced you that being FAIR doesn't necessarily mean that the data is going to be reproducible. Okay, so Fiona gave a little bit of my background as a neuroscientist, a vascular neurobiologist, and I now work as a Knowledge Exchange Fellow with Fiona. I thought I'd put her up there to acknowledge her, this fantastic person who is the Data Integrity and Quality Assurance Programme Director here at Bristol. Our main project is, we've entitled it Reproducibility by Design, which I'm just going to very briefly introduce. This initiative aims to embed greater levels of data integrity and quality assurance into research, thereby improving the reproducibility, the replicability, and hopefully the resulting impact of research findings. We train and collaborate with postgrads, with early career researchers and research groups across the faculty to embed best practices. The main inspiration for reproducibility by design was Joseph Juran's quality by design concept, which focuses on preventing defects in manufacturing through a risk-based approach to quality assurance. Fiona, with me then joining the team, adapted this concept for research processes. Now, I'm not going to go too much into that, but as part of the overall Reproducibility by Design project, we wanted to explore the possibility of running a pre-publication checking service. And initially, this started as a manuscript check for the inclusion of key statements, author contribution, ethics, conflict of interest, and acceptable data availability statements. and then doing an actual data availability check. Now, before implementing a pre-publication checking service, we wanted to determine the baseline level of the inclusion of these key statements in articles and the baseline level of data availability in articles coming out of life sciences here at Bristol at the time. So we assessed the statement inclusion in peer-reviewed primary research articles published in 2023, 100 articles, I should say, from life sciences. And while we found that conflict of interest and ethics statements were high, author contribution statements and acceptable data availability statements were lower, available upon request, unless it's fully justified, with instructions on how to request and access that data, is not an acceptable data availability statement. We then explored data availability further by actually seeing what was actually available. What could we find and access? Was the complete data set available? To try and answer this as best as I could, I attempted to find and access the raw data that underpinned all the results, figures, and tables. Now, I'm sure you can imagine that this ranged from being very straightforward and taking only a couple of minutes to it being me spending a lot of time digging around quite blindly, trying to match up data sets to figures. So I set a time limit of 20 minutes per article. Most articles, it was fairly straightforward what was what, so I could determine whether the complete data set was there or not. I did get a couple of articles, I couldn't work out what was what. Anyway, I'm happy to report that just under half, I was surprised by this, just under half of the articles we assessed had made the complete data set available. A small proportion had made only a partial data set available. This could be, say, just some of the raw data or some sequence data, protein sequence data, for example, or maybe some summary data. Then if you look in the grey section of the pizza, a quarter, 25% had made no data available with the available upon request statement unjustified, and then 17% no data available and no mention of data availability whatsoever. So basically, nearly half complete data set available, nearly half no data available. We noticed there were some big differences between the schools within the old Faculty of Life Sciences. For example, only 22% of physiology, pharmacology, neuroscience papers had made the complete data set available, versus 72% from psychological sciences articles. Right, so up to this point, it's been a case of, could I find the data set? And then could I access the data set? Could I download the data? In some cases, I did open the data and have a look around to check that if it wasn't easily apparent what, was all the data there underpinning all the figures. So this is a very basic F&A check that I had done with a bit of I because I had opened some of the data sets. But it wasn't a full interoperability check, if you like, and I also didn't look for reuse licenses applied at the time. So future iterations of this manual and basic fair check could include those elements. You know, could I open, modify it, and reuse the data if required? With my current computing environment, this would mean it's interoperable. And then was there a reuse license applied to the data? So the terms under which I could reuse the data were clear and obvious. Now, seeing as more than half of the data sets from 2023 in our assessment weren't available, more than half of the complete data sets weren't available. These, if you think about it, they would score a 0 in a FAIR score, so they don't even get, you can't even find them. So moving forward, as well as trying to improve the fairness of data sets, we also wanted to find out what are the barriers across the faculty to making data available. So before I continue with the fair checking, I thought it'd be interesting to share the results from a faculty-wide survey we conducted on the perceived barriers to data sharing. We surveyed active researchers from postgrads upwards to rate how often statements apply to them when thinking about making data available. We got responses from all career levels and from all schools across the now super faculty of health and life sciences. This is just a snapshot of the results, by the way. So I'm going to show you the top 8 statements out of the 21. No surprise, I don't have enough time to prepare my data for sharing was the topmost agreed with statement. Next came, it's too complicated, and I have too many data files. These, you can imagine, go hand in hand. The more data files you get, the more complicated it can become. Fourth was not having the rights to share, which of course is a legitimate barrier. This survey was across the super faculty of health and life sciences. So therefore, there are many health researchers that took part, and it goes without saying that not all health data, for example, can be shared openly. Fifth was the infrastructure is not in place, so technical barrier. Then came the cultural barrier of my team doesn't do it. You can imagine if your team doesn't do it, then there's no expectation for you to do it. So you're not going to set aside time to do it. And so therefore you perceive you don't have enough time to do it. Seventh, the knowledge barrier of not knowing how to share. And eighth, very large data sets. So we took away from this that the main barriers are the complexity of the data sets and the process of sharing, which leads to not having enough time to sort it all out, to prepare, and then to go and to share. And the findings have highlighted to us the importance of targeted interventions to enhance data sharing practices, and we're prioritizing data planning and management training tailored to each discipline, because planning to share from the outset could significantly, or should significantly, alleviate a lot of these challenges and barriers. Okay. So up to this point, I've introduced a manual F&A check. Now, I naively thought that on the most basic level, if you can find and access, i.e. download, and then open a data set, plus the reuse terms are clearly defined in a reuse license, then surely this data set is very likely to be computationally reproducible, or more computationally reproducible. No. Yeah, so don't get me wrong, being fair enables attempts to computationally reproduce results, but it doesn't equate to reproducibility. For data sets to be computationally reproducible, they need to be understandable or intelligible. Now, this may seem obvious to you guys on this webinar, but to me, a year and a half ago, it wasn't. Because if you can't understand it, if it's not intelligible, then you're not going to be able to understand anything, and therefore you might unintentionally misuse the data. You might not reuse it at all, and this would rather completely negate the entire point of sharing, and could even have and likely to have a detrimental effect, such as researchers wasting a lot of time trying to understand and reproduce the data. You can imagine the environmental impact of housing a load of unreusable or unintelligible data, etc. I'm sure there are more possible, negative effects. So it all comes back to effective data management, which is what we based the majority of our training on with PGRs. So, for example, use a file naming convention and logical folder structure. Detail and explain these in a README file so people outside of your research group can understand what's what. Describe variable names, exclusion criteria, missing values, and these sorts of things in a data dictionary. And if necessary, explain coding schemas in a code book. Okay, so how about we add an I for intelligible to fair, making fairy. Now, I've got young children at home, so I apologise. No guesses for where the inspiration for this came from. But in all seriousness, intelligibility has to trump fair. Fair is important, but the data needs to be intelligible first. Okay, so we trialed 3 pre-publication checking self-assessment services for researchers to use at the point of submitting manuscripts. All were operated in Microsoft Forms. One was on compliance with the ARRIVE guidelines. These are guidelines for reporting animal experiments. I'm not going to present those the results today. This one was a simple form asking questions about statement inclusion. And I'll just highlight, just some of the example questions here. The feedback received for this was positive, with ease of use 4.5 out of 5 and usefulness 4.3 out of 5. The other self-assessment form was set up asking questions on FAIR relating to the dataset, the research dataset. These questions were adapted from the Australian Research Data Commons FAIR self-assessment, and the questions align with the FAIR criteria provided by Mark Wilkinson and co in the original FAIR paper. Feedback for this was mixed, with some great feedback, some not so great. The main criticism was around the terminology used. And so the plan was then for me to go on and build guidance to go with this self-assessment to help people through the assessment. And also not just get through the assessment, but to also then go back to their data set and improve the fairness. However, the Australian Research Data Commons beat me to it. The original self-assessment was just a downloadable PDF, which is what I based the Microsoft form off. But then last September, they launched an updated version, adapted into an interactive web page that returns an overall FAIR score. And importantly, let me put it up. Importantly, this includes help with links to relevant guidance for each section. So instead of me trying to reinvent the wheel, we now signpost researchers to this form. And really, in my opinion, in our opinion, this is the best place to start if you want to verify your data when you're at the point of data sharing. Another resource I'd like to introduce is this fairly new resource from the French Elixir group. Elixir, if you've not heard of it, this is a pan-European network of bioinformatic infrastructure and expertise, and the network aims to support data science in life sciences. This is not a self-assessment. It's an automated checker. It's really easy to use. It's as simple as typing in the URL, URI, or DOI address and hitting the blue button, and it provides a score for each of the F, the A, the I, and the R, sorry, and then provides an overall score as well. It provides a breakdown of the score aligning with the FAIR criteria. Right, now something to bear in mind is this check is looking for the technical metadata that supports your digital object, so your research data set. In very basic terms, the metadata supporting the data set needs to be in a standard format with controlled vocabularies and recognized ontologies. If this technical metadata is either not present or it's not in a format that this checker can recognize, it will obviously return a low score. However, similar to the Australian Research Data Commons self-assessment, it links to guidance relating to the different criteria. So this FAIR checker really is checking how machine-ready the dataset is, not necessarily how human-friendly or intelligible the dataset is. I'm still firmly in the camp of making our research datasets as human understandable as possible, first and foremost, to aid effective reuse, and then make them as fair as possible, which should be straightforward if you've been able to make it as human understandable as possible, because that to me seems to be the significant challenge. Right, so I was then asked by Marcus Manafo here at Bristol and the editor of The Psychologist to assess the fairness of research datasets accompanying 30 articles that The Psychologist had featured. You can ignore these images on the left-hand side. The start of my article is on the right. So I conducted an article statement, dataset intelligibility and fairness check on 30 articles that were published last summer. Sorry, that were featured last summer in The Psychologist. So I assessed the inclusion of the key statements, whether the study was also pre-registered, the fairness of the datasets using the automated FAIR checker, and then whether or not I could make sense of the data. This last part was obviously using my very subjective grey matter. I was pleasantly surprised that the FAIR scores for the 30-yard scores came out pretty high, with interoperability slightly lower than the rest. But for the data sense check, this is very subjective, but I had strict criteria of marking. If there were only one or two aspects of the data set that weren't clear to me, and I would need to contact the author, and I was getting ready to start to computationally reproduce. So if I was trying to computationally reproduce, but I needed to check with the author one or two things, then this for me was a partial intelligibility score. If I was confident I could start the reproducibility, computational reproducibility, then that was I'd score that as intelligible. And I'm very happy to report that none of the articles, none of the data sets I couldn't make head nor tail of. However, these were psychology articles, and I don't, without wanting to sound belittling in any way, but generally psychology studies tend to be more simplistic in design, whereas you might only have one or two data sets. If you contrast that to, say, biomedical and neuroscience paper, for example, there might be 8 figures A to subpanels A to J with all different types of data file, data formats. And so trying to go through all of that and computationally reproduce all of that would be much, much harder. Right, so instead of just summarizing what I've just done, what I'd like to end with is with this Venn diagram. If a data set or a research output is fair, then it must be considered open research. So FAIR is at the core of open research. However, FAIR doesn't mean it is automatically reproducible. Rather, FAIR enables reuse and people to attempt the reproducibility. And at the intersection between open, fair, and reproducible research is a good old friend, effective research data management. And as boring as it sounds, we need to start showing research data management a bit more affection, because it is at the heart of our endeavours to produce high quality, reproducible and open research. Right, that's my last slide. We have a website, reproducibilitybydesign.com. We're also on LinkedIn. If you search for Reproducibility by Design, you can find out and get it, connect with us and ask us questions and come on board the team if you like. Right, that's me done.
Great, thanks very much, Nick. Just a reminder to everybody, you can either pop a question in the chat or you can scribble it down and we will have a Q&A session after our final speaker. So now I'd like to hand over to Dr. Catherine Winchester. Catherine has a PhD in physiology, pharmacology and neuroscience and is the head of Research Integrity Service at CRUK Scotland. Catherine is responsible there for fostering a positive research culture, developing and delivering training and research policies, and coordinating the Institute's research champions. She represents CRUK Scotland at national bodies such as CRUK, UK CORI, UK Rio and UKRN. In addition to all of this, Catherine also makes time to conduct detailed assessments of the research conducted at CRUK, which is going to be the subject of her presentation today. Over to you, Catherine.
Thanks very much, Fiona. And that was a really great talk, Nick. I was really interested in what you had to say. And as well as thanking, for the invitation to speak, I think it's been really useful sharing our practices and our conversations. And I think you'll probably see, Nick, that I've incorporated a suggestion you made to me about a year ago. So thank you for that. So I'll just share my slides. Okay, hopefully everyone can see those. Can you? Okay, great. So yeah, I'm going to focus on a pre-submission manuscript review that I conduct at the CRUK Scotland Institute. But I wanted to say it really isn't about policing, and it really forms part of what we've been trying to do at the Institute to enhance reproducibility by having this as part of our programme and aligning all our activities. So I've just got a few of them summarised on this slide. So being head of research integrity there, it's a small research institute. Pretty much everybody is a cancer researcher. The majority of people are biomedical researchers, but we do now have a growing body of data scientists, computational biologists, and modelers. And so one of the things that we do, as well as this manuscript review, is we have a mandatory research integrity training programme. And in order to help the reproducibility of, and the high standards of publications, one of the sessions is integrity in science communication and writing. Hopefully, Nick, you'll be pleased that we also run a data management training course. And actually, I think the checkers that you mentioned today for FAIR will be really useful to incorporate there. I also run manuscript writing sessions. I try and disseminate as much as possible. So I share information at the faculty. We have a body of research integrity champions, and they're actually based within each research integrity group, within each research group. And so that's a really great way of communicating and sharing practice, and actually having somebody in a research group that takes responsibility for this. So as well as the training, we have a research integrity toolkit, lots of guidelines, and now and policies as well. So we've been really prescriptive about the way we store and archive published data. So when people are conducting their research, it's up to them with some guidance how they manage their data, but actually we keep a copy of all the published data locked in a particular space on the server. And that's in addition to data being shared on a repository. And that's one task I have to do, is check every year. So it's a little bit of policing. And of course, we have lots of policies sitting around this. But today I'm going to be talking about this manuscript review. So it's mandatory. We've been doing it for about 10 years now. And it was really set out initially to make sure that we were publishing at the same high standards, so consistently across the institute, regardless of what data were being published, where the data were being published, that we just wanted to maintain some standards, meet reporting guidelines for our particular areas of research and particular field standards. And so whilst that's, the publication is the output, and the reasons for doing this were catching mistakes and errors, as well as, trying to increase accuracy and reproducibility and transparency of our reporting. But we really, as I said, wanted to align all this with other activities. And so I think it's really important that we aren't just saying at the end, oh no, you've done this wrong, or you should have done this in a different way. But we're trying to change behaviour and change research process, and recording of data and lab books, etc. And so it really has been embedded as part of our institute culture that we are very upfront and open about research integrity, but also the support and guidance that's available to our researchers. So moving on to the actual review process, a key part for me is actually people booking time for me to do the review. And so when researchers are nearing the end or getting ready for submission, they'll contact me, we'll arrange a date. And at this point, I share a publishing checklist with them that I've put together. And this is really a sort of one-stop shop. It's got all the information they need to know about the sort of peripheral things to do with the publication. It's to ensure that people actually use the right affiliation, they report the right grant codes. So it's all in one place. And then this is what I was referring to with sort of after discussions with this spot check group, that I've now introduced a questionnaire that researchers have to fill out before they submit their manuscript to me for review. And this is to, I suppose in a way, it's a self-assessment. It's got lots of key questions. I've got a slide, so I'll go through that with you, but it was really about sort of putting some of that responsibility back onto the researchers, because I found I was commenting on the same things across multiple publications. And actually, I don't conduct the review until this questionnaire has been completed. And the idea is that if people are answering no to the questions, that they really need to go away and answer yes and put in that information into their publications. I then get access to the completed questionnaire, the manuscript documents and the figures, and I conduct the reviews. I return the review as an annotated manuscript, as well as a report summarising the key findings and any actions that have to be completed. And as a little bit of a stick, we now will not pay the open access fees unless the data have been archived according to our policy and that the review report has been completed and returned to me. As a core funded institute by Cancer Research UK, we have part of our budget assigned to pay these open access fees. So we're in a position to control and have this stick at this part. So this is just a summary of the questionnaire. And as I said, it is about authors taking responsibility and making those considerations themselves. And so that by the time I see the manuscript, I'm either advising or just commenting minimally on these things. So the questions do relate to best reporting practices, some of the standards that we have introduced at the Institute and expect people to comply with. It's based on my experience as a researcher, looking at field standards, and as I put on here, looking at reporting requirements of journals such as Nature, Embo and Cell Press. Journals that we quite often publish in, but I think they've got some really good reporting checklists, particularly for biomedical research. So just as an example, on the questionnaire, I've got some questions about experimental design, asking people about, fairly basic questions about their replicates, to consider what statistical analyses they've done. Are the data available? Is the code available? And where are they? What repositories and have the access IDs been included in the manuscript? There's quite a few questions about methodology, but also in terms of sort of reporting, making sure that we've got clarity. And so in particular, graphs. So we have asked at the Institute that people don't just hide data in bar graphs and not explain what error bars they use, but we actually want people to make their really think about what they're trying to convey in their graphs and show the data points and actually describe what data are being presented. And so that it's really clear from the figure, but also in the figure legends as well. And as I said, this has to be completed before I do the review. And so if it isn't completed to a satisfactory level, I send it back. And as I said, if there's lots of no's, then again, I engage with the authors to work with them to try and understand and help them make sure that they can answer yes to the questions. So the actual review consists of 4 elements, and our manuscripts, and these are on the left-hand side of the slide, labelled as CRUK SI manuscripts. This really refers to papers from the Institute where we are first author, corresponding author. We've generated the majority of the data. And these manuscripts go through the full 4 processes or steps. We also write a lot of reviews. They tend to be narrative reviews rather than systematic reviews. And so they just have a text plagiarism check. And if there's figures, then I put those through an image twin analysis check. We post a lot of papers on bioarchive, so we have a lot of preprints, and because of the volume of the work with the manuscripts, the time and the number that I have to review, we realised that there was sort of a bit of a bottleneck there. So for our preprints, they're just going through the first 3 steps of the process until they get submitted to a journal, and then I'll do the full integrity review. And then likewise with the collaborative papers, and we have a large number of collaborative papers. I don't have time to fully review those. So again, I will just do the first 3 steps. And actually, I want researchers and authors to take on some of that responsibility to I suppose, essentially to conduct some of the checks that I'm doing. And that's where the questionnaire is also really useful for them to actually start looking to see how the data presented by other groups or their data represented in other people's papers. So my next few slides are just going to go through a little bit of detail about these 4 steps. So for the text plagiarism check, I use Authenticate. It's the sister product Turnitin, which I think pretty much all universities use to check undergraduate work. Journals and publishers use Authenticate. So you need to buy a license, and there's a cost per article. And so you upload your text, and it screens across its vast and ever-growing database. And the software checks for word plagiarism, which I suppose we're really concerned about, that people aren't just literally copying other people's work. You can also tell whether people have paraphrased rather than actually writing in their own words. It's useful for checking self-plagiarism, which often is breaches of copyright where people have reused their methods and they've already published a method and actually they don't own the copyright anymore. And most recently, Authenticate has introduced a new feature, which is an AI writing indicator. I haven't tested it yet, and nothing I've put in to Authenticate has come up as being AI generated, but something that I'll have a look at and maybe test with some ChatGPT text that I create. So yeah, it's really useful for checking for obviously plagiarism and cheating, but also making sure that researchers aren't breaching copyright. The article information check actually covers some of the things that Nick was talking about in his presentation. So sort of peripheral, but obviously really important information to making sure papers are reproducible and honest. So I look at the author list and check authorship criteria. I check affiliations to look for accountability and actually who's responsible for the data that are actually in that paper. I check to see whether there's a contribution statement and that all authors' contribution has been included. Also funding acknowledgements, both for the research itself, but also the authors. I look at the acknowledgement section to make sure that that's been, people have been appropriately acknowledged. I then check the 4 statements and read the statements on competing interests, data and code availability. As I said, we've been really prescriptive about the published data. So then I also ask a question on that questionnaire about have they collated their data and have they started organising it. And as a research institute, we have a lot of advanced technologies and services. And so data from these groups goes into the paper, a lot of our papers. And so again, it's really useful to check that the methods have been checked with the leads, the data are presented and have been analysed appropriately. So that's sort of what I class as the article information check. We purchased a license for an AI forensics tool about a year ago. So we're using Image Twin. And I have found this really useful. So it has a database, as you can see, of 75 million published figures. The database is growing. I find it really useful for checking for duplications within manuscripts as well as across the database. I think, the database is limited because although it's large, it doesn't have every published paper. So it's possible that you're going to miss a duplication across another publication. But I've just got some photos on here where Image Twin has picked up 2 pancreatic cancer sections that were used twice, both the one in the pink box and the one in the green box were used twice in the same paper. So you can actually see, if you look at the green box, it actually, because it's doing pixel recognition, the image doesn't necessarily have to be the same. And things like that, and we do a lot of Western blotting. I don't know if that's familiar to people, but that's really difficult for a human eye to pick up, particularly when you've got panels and panels in a paper. And again, Image Twin have just added an AI generated content searching tool. And so far I haven't come across anything in any of the papers that I've looked at. There are also lots of false positives. So it does, I think, you do need to go in as a human, understand the data and have a look. For example, the serial sections of a piece of tissue, that the tool will pick those up as duplications. Recent features that were added to the tool were sort of a confidence anomaly detection. So it also detects manipulations. So if images have been enhanced but only in a particular area, or cut and pasted, or it will pick up things like that. But it gives you a confidence rating, which you can usually tell by eye, but it is quite useful to have a look at as well. And then this slide actually summarises an entire day's work. So I do spend a lot of time scrutinising manuscripts. So it's not peer review, although I am a biologist. I'm not an expert in every aspect of research that goes on across the institute. But I read the abstract and introduction. I expect as a scientist that I should be able to understand it. It should be clear and accessible and informative. But in terms of reproducibility, and again, I agree with Nick, we're doing a lot of these activities to ensure or to help promote reproducibility. It doesn't necessarily mean that just because everything that I'm saying here is right, that the data are going to be reproducible. But we are trying to be transparent and as accurate as possible. So with the rest of the manuscript, I take a lot of care and attention reading it. So the results section, I tend to read sentence by sentence, and each finding I then check that actually what is shown in the figure panel is actually represented in the text and vice versa. And that the figure is actually really informative and clear and accurate. And also the figure legend aligns with that, and you can interpret the data. I look at the figures, of course, as I've just mentioned. I'm looking at the accuracy of presentation, the quality of the data, and whether the way they've been presented complies with particular field standards and the way we would like people to present data at the Institute. Figure legends, yep, I've mentioned that. So your standalone detailed information. I spend quite a long time reading the methods, checking the methods, and checking with other people that enough detail is in there and that we comply with our field standards. So as well as using Image Twin, I still check the figures by eye, and I look for duplications. Image Twin isn't very good at looking at data on graphs, so I scan each graph by eye and look for reuse of data as well. But so when I do the full integrity review, this is what takes me the day per manuscript. And then returning it to the authors. So as I mentioned at the beginning, I return a report, which is a summary of what I've found, including feedback or information from Authenticate and Image Twin, and a list of actions that must be completed. Also suggested actions, and as you're reading a paper, it's really hard not to proofread and edit at the same time. So I often make editorial suggestions, and I return with the report the manuscript, an annotated manuscript, and an annotated document of the figure legends. So that's really about the process. And there are some challenges. It's not a perfect process. I think the institute where I work has grown, so with the number of publications is rising. And so actually that's quite a challenge to get this work done. Other challenges I found are incomplete or incorrect questionnaires. So people might have answered yes to something, and then when I read the manuscript, I realise, I don't know, they've just said yes, or they haven't understood. The recurring mistakes is an issue, but actually by being involved or delivering the research integrity training and writing the policies, it gives me an opportunity to identify practice at the Institute, try and make changes, and try and make changes early on by incorporating that into training and guidance. Obviously, I find there's missing information, and the next thing I was going to say is, all authors checking the manuscripts. That seems to be a major issue, that often people get to the end point and they're really, excited to submit. And we find this also with our collaborative manuscripts, that we get sent, an email with a paper saying, oh, here's a paper, I'd like comments back within a week. If I don't hear from you, I'll assume that everything's fine. So I think it's really key that all authors read the manuscript, particularly the bits that they've contributed to, because they are responsible for that. And I think, you know, that way we will minimize mistakes. Another area that I've noticed is that mistakes creep in compiling the final versions or at resubmission or revision stage. So I'm beginning to check more of those as well. So we've had a couple of cases where I've commented on a manuscript, identified things, and then other things have crept in as the authors have incorporated other comments or they're finishing off their paper. Yeah, the archiving of source data. So that's been policy for about 10 years, and I'm still struggling to get people to comply with that. So it sort of sits between 50 to 75% of people are complying with our archiving on our server and putting data into repositories. And then this is my last slide, because I've been asked this a few times. You know, what could be done at scale in a multidisciplinary environment? Because I appreciate that I work in a small organisation and, you know, we're really focused on cancer research. So I think, you know, organisations could have their own publishing guidelines or departmental guidelines with specific checklists. And I suppose, you know, the FAIR self-assessment is a really good example of this. I know some groups are doing pre-submission spot checks, or whether you could do reciprocal checks and offer peer support. And I think training is key. And I've just introduced writing clubs in addition to the training. So I think that will help to remind people and refresh people's memories of some of these issues. So that's the end of my presentation, and I'm really happy to take questions later on and look forward to hearing Mark's talk.
Lovely. Thanks, Catherine. I also should have said, as well as the other very busy things I said in your bio, you're also the UKRN local network lead for CRUK as well. So as if you didn't have enough to do already. Okay, so now we're going to move to our final presentation from Professor Mark Kelson. He's Professor for Statistics for Health at the University of Exeter and is a self-described data botherer. He's an institutional co-lead for UKRN and has a background in clinical trials, but increasingly works in data science and has interests in causality, physical activity, and policy evaluation. Over to you, Mark.
Just getting off mute. Okay, hi, thanks everybody. Really inspiring talks. I love the idea that there are people checking this stuff. I love the idea of slow science, us taking our time. I remember seeing a tweet by Kathleen Moran on Twitter years ago saying, you know, she loves science until she goes and reads a scientific paper and then she finds it really boring. And it's like, you know, that's what it is. You know, people have this view that science is this really flashy, you know, big splash news kind of thing. And it's not. It's incremental. It's people doing hard graft. Doing things properly means doing them a bit more slowly. And I love that we have this focus today. Okay, so I'm going to talk to you a little bit about a reproducibility evaluation we did at Exeter. But before I do that, I'm going to give you a questionable anecdote. Hang on, let me go on to my next slide. This is the major issue that I think that I'd like you to take away from this day. When you board a plane and you're about to take off, there's a number of things that you're asked to do. Put your tray tables up, put your seatbelt on, turn off your phone, put your blind up. And some of those things they check. They do check your blind. That's so that if it crashes, they can look in and see if there's somebody in there. They check your seatbelt. They do check that your tray tables are up. These are all critical safety issues. They do not check your phone. Now, what does that tell you as a passenger? It tells you that it's not important. It's not an important thing. There's no evidence that phone signals interfere with the operation of a plane, right? And that's why they don't check it. So this is the signal that you're being sent as a passenger. The only issue is that your phone is going to try and connect to networks and it's going to run out of battery. That's basically why they tell you to turn to flight mode. In a supermarket, they don't check that you return your trolley. It's a societal good, but the supermarket will continue functioning if you don't do it. And they do incentivize you a little bit. I think the same is true of reproducibility checks. If we think reproducibility is important, we should check it. Because if we don't, we are sending a clear signal to the research community that it does not matter. Right. That's the main focus. And if you're trying to convince everybody here knows reproducibility is important. But if you're trying to convince somebody, I think that's an argument that I think does hold a little bit of water. We tried to do something similar, on spot checks. We stopped calling them spot checks fairly early on, but it's based on this idea from the Sainsbury lab, where they do lots of plant science research, lots of original data collection. They have a lab of people and they select 3 figures from 3 manuscripts every 3 months, and they check the provenance of those figures or tables in manuscripts. So that's what they do in the Sainsbury lab. We tried to do something similar, but there's a few differences. First of all, we wanted to do this in disparate disciplines, not just plant science, not just with researchers that we knew about, not just people doing original research necessarily. So we chose 3 departments from one in each faculty, chosen by faculty leads. And we use the risk-based strategy that Fiona is a proponent of. The most important thing, you should check the most. And so we decided that we would look at quantitative claims in the abstract, because we figure that if it's in the abstract, that's the most important thing. We are not checking Appendix 3. We are just looking at numbers in the abstract. Are those reproducible? We're looking at published output. So this is a post-publication check. I'm going to come back to that in a minute. And again, like others have said, we're a very fuzzy kind of group, I think, that there's no punitive element. When this came to me to begin with, there was a little bit written up on it, and there was kind of a yellow card system, and there was going to be a report sent to your head of department and things like that. And we felt that this could just be entirely supported. And so we piloted this last year, and I'll give you an idea of the timelines briefly. I'm going to try and rattle through this so that we have a chance to chat at the end, but there'll be time for questions. There are some benefits to this. So when I first, Fiona mentioned that this is a couple of years in the making. I presented on this way before we ever attempted it, and got lots of feedback saying this was not a very good idea, and it wasn't going to work particularly well. And lots of that feedback was really, really helpful. But what I felt was that I hadn't really enforced the idea, what are the benefits? Why would you do this at all? And there's some really good benefits. This is a light touch approach. Randomly selecting from papers in a department means that most people are not going to be selected for this reproducibility evaluation, but they all know that they might be. And so it kind of raises standards. People are aware that one of their papers might be selected. We view this as a teachable moment. People love talking about their own papers. And so if you can give them some advice about how to make their own paper more reproducible, I think that is really a good time to talk to people. They want to hear that, and it's tailored to their own work. So I think that is quite nice. I think in Bristol, they're talking about trying to show this to external funders and say, an idea of we are doing these checks, and so you can trust the work that comes out of our university. And I think that kind of thing is useful, and it does motivate reproducibility at all levels. The burden of reproducibility tends to fall disproportionately on junior researchers. They're asked to do more and more and more. They're the ones in the job market. They're the ones who are sort of bearing this burden, and we're asking them to do more and more. And I think this is just something that means that everybody is motivated to be reproducible. So this all happened in a blur last year. We were told, I think it was February 28th, that the project was funded. We planned it in March. We hired in May, ethical approval by June. And then we only had 2 months to do the thing. As ever, the scientific work was squeezed towards the end because it took a while just to find the people. And ideally, I would like a bit more time to plan this if we were doing this again, which I would like to. This was the team. I'll just tell you a little bit about them. The people on the left are the reproducibility evaluators. They're the people who did the actual work. And so those were all recent data science master's graduates of different flavors from the University of Exeter. And they contacted me through LinkedIn, or I contacted them. And they were just, they happened to be available at short notice at the time. We did this through a casual claims process. So we didn't have to advertise, there was no time to do that, and the contracts were short anyway. And so these were all vaguely science-related, engineering, computer science, and general science, I think, were their undergraduate degrees. Chris Tibbs is a librarian at the university, and he was very helpful with collating the outputs. John Luff is a senior research software engineer. He was good to help with queries about coding and things like that, and he helped coordinate. And Eilish, Gavin, and I are the co-institutional leads for reproducibility at Exeter. So that's the team, and I'll talk a little bit about the things you might want to put in place if you were trying this at your own institution. And I would like to talk to anybody who wants to try this at their own institution. So the three departments we chose: economics, social sciences in Penryn, and biological sciences in the EC faculty. And so they're all sort of slightly different sizes. My thing is just obliterating key figures. Yeah, so economics is 43. Haas Penryn had 13 papers. These were all selected in the calendar year 2023, and the clinical and biomedical sciences was the biggest producer of outputs. So we decided we'd select 12 at random from each of these three, so that we could have a random element from Haas, giving us 36 articles. And the long and the short of it, I'm not going to labor these numbers. This is a small pilot. I don't think this is the finding, really. The finding is the process evaluation work that we did. But I will just go through it briefly. Roughly half had statistical claims, and I'll talk a little bit about how we operationalize that. Of those half with statistical claims, roughly half had data available and less than half had code available, and about a quarter had both data and code available, and those were our sample on which we could conduct a reproducibility evaluation. Of those quarter, those 4 papers, half reproduced, 1 didn't reproduce, but might have done with more time, and 1 again didn't reproduce within the time frame. I would also say we didn't contact authors, so we didn't ask for any help. We wanted to do this reproducibility evaluation without resorting to contacting the authors. And you can see the reasons for no statistical claims. There's lots of qualitative research within that. I recently presented this, and I got a query about, of those with statistical claims, we should do a further dive in to see how many of those where they didn't produce code, where they didn't produce data, were there valid reasons for that? What were the reasons? And I haven't had a chance to go back and do that, but that is a work in progress. Okay, next slide. So one of the things that's interesting is, what is a quantitative claim? So we had an idea in our head about what a quantitative claim would be. The highlighted bit here is from a qualitative paper. There's a range calculated, there's a median age reported, there's a number of people, number of patients, and a proportion or number of males. We did not view this as a quantitative claim. So we did not conduct a reproducibility evaluation on this qualitative paper, but others may disagree. Our view was that this was not central to the finding of the qualitative paper. This is not, even though it appears in the abstract, this is not critical. This is not the final conclusion of the paper, so not as important. And so we didn't, from a risk-based perspective, we didn't view this as the main claim, and so we didn't view that as a quantitative claim. But perhaps we could have been a bit more explicit in our definitions at the outset. And so I'm gonna we did the evaluation. We selected the papers, but we also contacted the authors to see if we could get some feedback from them on the process. And so that was a little bit confusing in that we asked them for their consent to send them a questionnaire to ask them about how they found the process. It was not a consent form to say, do you consent for your paper to be included? Because we felt we could do a reproducibility check on anyone's paper at any time, and they don't need to consent for that to happen. But that confused a little some people, I think, and we would want to clarify that. I highlighted one view here that this scheme duplicates reproducibility efforts undertaken by journals, and it is not my view that is universal. I think that's unusual. I don't believe that lots of journals are conducting reproducibility evaluations. I don't believe that the review process typically involves downloading data and code and rerunning analyses. Maybe in some fields, and I know some computer science journals have got badges of reproducibility, but I don't think that's a universal view. I've left out some other stuff just for time, but I do want to highlight this just to remind everybody, like everybody here is interested in reproducibility. We're all fighting the good fight. We're all on the same side. That is not the wider perspective at your institution. I feel sure of it. I'm sure this is the case across the UK. There's pockets of excellence, but there's also the opposite, right? There's pockets of people who are resistant to these efforts. And this was a senior academic who responded to our reproducibility evaluation. Their view was that the paper that we'd chosen, it was completely inappropriate. It was a small study and there was just simple statistics that were done. I looked up this paper and I don't share this view. I think there was some statistics that were done in there that could easily have been checked. And it does worry me. My view is that if your paper was selected for a reproducibility evaluation and you thought it was you thought your paper was so simple that it would be 100% reproducible, I think you'd be saying, yes, pick me, definitely take that paper. And if you were a bit worried, you might respond like this. And so I will say no more. A bit disappointing. So the takeaway from this is the people that you're talking to about this work probably are not as passionate about it as you. And you probably, there's work to be done to win hearts and minds in the reproducibility effort. And I think it's always worth reminding ourselves about that. The executive summary of what we found. Participants were broadly supportive. This was light touch, like they basically didn't need to do too much. We tried to do the reproducibility evaluation without their input. Some people felt that they wanted more notice, but really there was nothing that was being asked of them. So, I think there's no particular need for them to have lots of notice. Some felt that they would like more control over which paper of theirs should be selected, which I think is a kind of a misunderstanding about what a check is. You don't get to pick the homework that gets checked. It's a random check, and that's the point. And there was mixed views on the usefulness to the researcher. And so there was some people who expressed the view that if errors are found post acceptance of a manuscript, what good does that do to the researcher? And I think there's, from a utilitarian perspective, that's true. But from a scientific perspective, I think that is the antithesis of science. If, errors that are found are important at any stage. But I will come back to the timing in a minute. I have a reflection on that. This is the kind of thing that we gave people, supportive feedback. It wasn't just this. We didn't just go straight into this. We kind of said, thank you very much for your paper. Your paper was great. We really like these bits. But then we did give specific, tailored feedback to papers for how they should be improving their reproducibility. And what I would also like people to take away is, but reproducibility is a spectrum. So I was, interested in what Nick was saying. He gave himself 20 minutes to find the data, I think at one point. And that's important, right? You need to know under what conditions is something reproducible? Do I need to spend weeks at this and then it's reproducible? And does that mean it's reproducible? If somebody hands me a box file of paper questionnaires, I could technically type them out. I could technically reproduce that, but is that reproducibility? And should a random person that we drag off the street be able to reproduce your analysis? Or should they have to have a certain level of expertise to be able to do it? What is the resource input that's required? Should the 17th decimal place of the third table in Appendix 7 be reproducible? It's a spectrum, right? This is not a binary thing. And I think that is something I'd like us to all to reflect on. And the time scale is really key as well. Nothing is reproducible in perpetuity. Code rot will destroy all of our work. So some of the reflections, and if you're interested in doing this, I'd love to have a chat with you, but these are some of the things. Senior leadership buy-in is important. Responses when there was a prof on the email chain were qualitatively different to when a prof was not on the email chain. And that is a regrettable feature of some aspects of academic science, but it is the truth and the reality. And so if you're trying to do this, get some senior leadership buy-in and lean on them. And I think that will help. You'll get a different nature of response. I employed people on a casual claims basis. That meant they didn't have fully affiliate institutional access. So any institutional software that we have, they couldn't download. So we had some issues with Stata. So we have an institutional license, but some of our reproducibility evaluators, they didn't have access to it, and that was a pain. I'd love to have a budget for data access. So there's lots of biobank data where they say the data is available. You could go and reproduce it, but we didn't have budget to access that data. And so that would be a nice thing. Terminology is key. A view was expressed that the reproducibility movement is gendered, and that just the very term reproducibility was off-putting to certain disciplines, and open science or open research was a more inclusive term. And so I think we just need to be careful about our language and try to make sure that we're taking everyone with us. Like I say, there's work to do to win hearts and minds. We recently did a survey across all departments in Exeter about views on preprints. We just wanted to know, what is the cultural view on whether preprints are a good or a bad thing for people's career? And at least one department came back saying, what is a preprint? And so, it is not the case that everybody knows all about aspects of open science and research culture. Second last one is a bit embarrassing. We did this in a rush, and when I was writing this up, I was pulling together files from different places. This was all my fault. I didn't do enough training of our reproducibility evaluators and give them the tools to all do things similarly. But it was a struggle to check the numbers, and I think our own work on this could be more reproducible, and if we were doing it again, we would ensure that it was. And like I say, our consent process was confusing. Next steps for us. One idea, which I think is nice, is to focus on ref returns. So ref has a lot of effort associated with it. Every university is putting together their ref statement, and they will have a bit on research culture. And they will have a grid of potential ref returns. I wouldn't mind getting access to that for our institution and checking those. Those articles should be reproducible. They are the finest outputs that we have. And I think that you get a bit of buy-in. Senior management are keen on getting your ref return to be good. And so I think that could be an open door, and I'd like to focus on that a bit. Lots of reproducibility checks happen pre-submission, and I get it, right? That's the point at which things can be fixed and changed. And that's really, really good. And for a long time, I felt like, maybe our reproducibility evaluation had missed the mark a little bit. But there's a few good reasons why you might want to check stuff after publication. And first of all, the timing is a real key. People, you don't want to delay people when they're trying to publish. It's already a long process. If you do your reproducibility check pre-submission, and then a reviewer comes back and asks you to change something, in theory, you should go back and restart your reproducibility check all over, and it could get very bureaucratic. And so there's two good reasons. But the third good reason is the point at which stuff is most likely to be accessed for reuse is post-publication. It's not pre-publication when people can't see it for the most part, unless it's pre-printed. And so the time that it's released into the wild, that is a very good time to check whether it's actually reproducible. You can at that point check to see if in a reasonable time frame, if code rot has happened, if packages have been updated. That is the time when people are going to be reusing it. And so I think that is a good, in some ways, that is a very good time to be checking. We'd love to do this with more disciplines, so we chose 3, but I think there's lots of work to be done in other disciplines. One nice idea is potentially as a secondment opportunity for PhD students. They could take a 3-month pause in their studies to do reproducibility checks on articles in their field or in their department. Gives them a leg up in cutting edge, by definition, publishable quality material, and I think is probably an eye-opening thing for them anyway. But it did cost us in or around 20 grand to do this, and I think there was inefficiencies. We could have been a bit more slick. I think if we were to do it again, it wouldn't cost us 20 grand. We've done a little bit of the groundwork, and so we have some processes. So I think there would be, if we were rolling this out at scale for longer, it wouldn't be quite so expensive for each check. Okay, I am going to pause there, to stop there, and give some time for discussion. Stopping sharing now.
Great, thanks very much, Mark. So while everyone just thinks about any questions they might want to put their hand up and ask, I'm just going to go back to one in the chat, which was asked by Matthew J. But entirely agree, data management is essential, but researchers don't usually have experience or training of it. And then the typical data management courses like creating databases and SQL are usually too broad for what we need. So the question to you, Nick, are undergrad or postgrad courses the right place to get more data management training, or should this be done elsewhere?
Well, good question. It depends on the particular discipline, the course, the postgrad course. We have been working on this project now for 2 years, and we have put together core modules that cover the basics for data management, and now we're tailoring those to make discipline-specific training modules. So it really depends on the requirements for that particular course and type of research. We're developing, to give an example, we're now developing guidance around how to anonymize and share qualitative data, which is something that we hadn't really considered, a year ago. So it's keep developing, keep refining, and making bespoke training is what I suggest.
I guess one thing I'd just like to add from my involvement is that it's not just postgraduate students or early career researchers. I think people at all career levels in many disciplines that we've worked with have appreciated some training because as Matthew points out, this is generally not part of either an undergraduate or postgraduate structured program. So I think our key message is that everyone can benefit from a bit of this training because the tools are moving on as well. The fair checks are moving on. Repositories are moving on, so we all need to keep up to date. Thanks, Nick. I'd like to hand over to Akira now, who's got her hand up. Please ask your question, Akira.
Am I audible?
Yes, we can hear you.
All right. Well, thank you. Thank you to all the 3 speakers. I learned a lot. This is one of my favorite topics to think about alongside my research. So this was great. Thank you. I'm going to start with a question. I've got 2 questions, one specifically for Mark, and then the second one for anyone who wishes to answer, really. So Mark, I really like your presentation and everything you covered. I think it's really important. You said something about the idea in the very last part of your talk. You mentioned that it's worth considering looking at these reproducibility aspects post the publication or after the piece of work is out there. And I think that's a really interesting perspective because you're right, we try to look at all of the different things, your code, your OSF, all of it before we submit our work, but it's definitely worth looking at that perspective. A quick follow-up on that is that considering, hopefully there isn't a problem, but if something does come up in our work post the publication or post the work being out there, what do you think we can do? Because as I understand, the process of getting work out is a bit linear. So if you've got the approval from the journal, then you accept it and it goes out and that's it. But if he has such checks, which I completely agree is super important, and if something comes up, what do you think needs to change in the publication pipeline? Because how does that look like? Is it that we can kind of take things back, make some amendments, kind of make that as a public note that, okay, we have changed certain things post the publication because of these reproducibility checks or something like that?
Yeah, I absolutely think it's a great question. So from my view, I think if we find something is not reproducible post-publication, there's a few options, but one is an addendum to the paper can be written if it's a really big error. But secondly, if there's some good practices being done and there's a link, for example, to an external repository, that can still be edited. That can be improved. That can be, you can have version 2 on the basis of some reviews. So it is not so much the case anymore that the final output is completely unmodifiable, given how we kind of link in other resources. So, I think there's still things that can be done to benefit if errors are found post-publication.
Right. Thank you. Right. And my second question actually stems a little bit from the work that I do. So I work with a lot of field data, and all of my PhD work follows sort of the open science principle. So we pre-register, we've got all of our data accessible, and everything is out there. But a part of the work that we do is also with data that is proprietary in nature. And I'm kind of heading towards a project where now I have to be working with proprietary data. And I was thinking about any of the three of you, if you've got any comments on how we can go about making or working with such data in the context of open science, or the FAIR policies that Nick covered, for instance, at the very beginning. If you've got any ideas, yeah.
Do you want me to come in there?
Yeah.
Sorry, the audio cut out for some of that, so I think it's my Wi-Fi here. I only heard half of that. So sorry, just so your question is about the challenges handling proprietary data formats.
Yeah.
Well, I think as Mark alluded to, it's a spectrum, it's a continuum. We're trying to make our data as fair as possible. So there's only going to be so much you can do. Some proprietary software, you can get it to output in, say, a simple text format. I don't know how useful that would be, because you might need the proprietary software at the other end to turn that back into the proprietary file format. And so it all depends on the software, but there's only so much you could do. So look into, investigate what you can do with your proprietary file format. What can you convert it into? You can have proprietary formats that are actually open formats. So, for example, Microsoft formats are OpenXML, and that means, on an Apple phone, you can open a docx, you can open an XLSX. So, although it's proprietary, it's not a barrier, because it's an open format. So
Right.
it's not black and white. It's a graduation. So try and investigate what you can do to make your data as fair as possible.
Mm-hmm. Yeah. So far, we've worked with that particular data set for most of the chapters that I've worked on, but the way that I went about it is to derive summary data sets, which is what was then used for the modeling and the statistics in the papers itself. So those are the ones that I've made available, but the actual base data from which it's kind of generated, I've only been able to give a metadata in OSF along with all of the other files that, okay, this is what we have. But now we're getting to a place where we have to work with the raw data, so we can't really take the summary out. And so I was just wondering, how do we go about making it accessible despite sort of this very important barrier? But yeah.
But is it so? Is it sensitive data that you can't share, the raw data?
Yes, indeed.
Yeah. Okay. So you've done as well as you can there with it sounds like you've done as well as you can by making a metadata record describing the raw data, and then you're sharing summary data as well.
Yes.
You could potentially look
in
at
other projects, though not this very project, but in other projects we've got summary data sets, yes, of the raw data.
Yeah, you could potentially look into that. There are synthetic data sets that people can generate that mimic the raw data set. So then people can handle those synthetic data sets without handling actual, the real sensitive data. I don't know if that's something that you could explore. But again, there's only so much we can do. As long as we're pushing to do as much as we can, then that's surely better than no data available, etc.
Absolutely. Thank you so much.
Can I also, to go back to your first point to Mark, if you look at the open peer review, if you look at some open peer review journals where the article is accepted and then published and then peer reviewed in an open format, you will tend to see, if revisions are made, you will see revision 1, version 1, version 2, version 3, and it's all transparent, and you can see the comments and the counter arguments, etc. That sets a precedent really for me with how we can, once something's published, it shouldn't be set in stone. If people find errors in your work, well, I think we should see it as owning up to those errors and saying, hey, look, we've made a change here because of this, something we didn't realize.
Yeah.
The fact that people have owned up to it, to me, is like a badge of respectability more than just hiding it or not doing anything. So the open peer review model with the versions, etc., that seems to how we can move forward in these challenges.
Thank you so much. Thank you.
I'm going to hand over to Mark now.
Just briefly, I heard about a situation where a researcher at Exeter was working with some proprietary data, and the data owner refused to make it open, but they successfully negotiated with that data owner to randomly select 5% of the data and make that available so that people could check that the code runs. And I thought that was quite a novel solution, so that at least people could check the reproducibility of that the code works in some sense. There was some check possible.
That's interesting. Yeah. Thank you so much.
Right. Any more hands up? Otherwise, I might turn to a question in the chat. I can't see any. If you have got your hand up and Zoom isn't showing me, then please shout. Okay, so I'd actually like to revisit a question from Elena in the chat. I know, Catherine, you have provided an answer, but I think that this is something that is actually worth sharing with everybody. So Elena's question is, was it difficult to justify your position, Catherine, and the entire research integrity service to the organisational leadership? And how do you measure that this service is meaningful and impactful?
So the answer to the first part was no. The job was created by the senior leadership. I suppose I was left to develop it, and I really didn't want it to be about policing. I wanted it to be about training and fostering. So I've worked quite hard to make sure that I'm approachable. We've got rigorous policies and practices in place, but actually that it is all about being fair, being honest, and getting the best for everybody, and dare I say it, for science and research. So I really made that quite a feature. And actually, I was just going to add to what Nick was saying about the mistakes. So another area that I've worked, I do support people post-publication. I don't screen every paper from, before I was at the Institute, but I do, I search PubPeer every year and look for all the papers at our Institute that are on PubPeer. And then I work with the researcher where we've got the data to correct their papers. And actually it's viewed really positively because they're not judged. It's not a misconduct investigation. It's looking at the evidence, it's looking at the data and then doing something positive so that we correct the version. So I think so no problem with senior leadership. They wanted the role. They're very happy with, our outputs and what I've been trying to do. So actually the director endorses everything that I do. So I said I go to faculty meetings. That's because he keeps inviting me to come and go and talk to them, tell them what you're doing. So I think that really helps. I'm sure there are people that, Nick, you said, Mark, you said about, the time and potential delay to publication. That I don't really get that negative feedback, I think, because I try and work with people to arrange a date that suits them. And so people value the fact if I pick up a mistake before their paper's gone out, I think, it's less embarrassing. So yeah, it's all very, very positive, actually, very positive. And the second part was about metrics. So I don't have any metrics. I don't measure it. I suppose it's written and verbal feedback, the fact that people openly come and talk to me. So actually, as well as me screening PubPeer, people voluntarily come with problems with their papers and are quite open and say, oh, look, this appeared on PubPeer. I know you haven't found it yet. Can you help me? So I think, the feedback that I get verbally and written is probably the only metric that I've got, and it's all very positive. Does that answer that second part? Don't know if the person.
I think so. Yeah, thank you. So we've got 1 minute left. I don't know. Maybe I'll just hand this back to Mark. So Mark, this is you created this webinar, and you've reported on your work. So I wondered if you had any closing comments for us. Well, I really enjoyed it, and it's furthered my understanding. But I'll just give the last word to you.
Goodness, not prepared for that. But what I did like to see was that, you know, there is a community of people here, and so, you know, we are borrowing each other's ideas and extending on them, and I think that's as it should be. For people who want to get involved and want to try some of these things at their own organizations, I think we'd love to hear more of that, and we could, you know, combine some of these things to make a super duper check for reproducibility at your institution, and you would own it, and it would be your thing. And you know, this is not about you know, it could be your idea basically. So we'd love to chat about that.
Great, thank you very much. So yes, one final thanks to Nick, to Catherine, and to Mark, our speakers for today. Thank you for UKRN for hosting this webinar, and to Deanne as well for all the logistical side of it. And I hope to see you again soon. Thank you very much.